So we're going to talk about active processes in this part 4 of chapter 3. So active processes require energy in the form of ATP, and usually they move molecules against their concentration gradients, or otherwise known as uphill. Primary active transport, this is the process in which solutes are moved across cell membranes against their electrochemical gradients, and this is by using the energy supplied by ATP. Here we have the example of this protein, the sodium-potassium pump, or the sodium-potassium ATPase, and this is a really important example of primary active transport. So here in this example, we have the hydrolysis of ATP, which results in the phosphorylation of the protein. So here ATP interacts with the protein, it turns into ADP, and it leaves that phosphate group attached to the protein. And what that does is that it changes the shape in a manner that kind of pumps the solute across the membrane. So here we have the binding of ATP causes it to go into this V shape, open to the extracellular fluid. And when it's open to the extracellular fluid, this is when potassium can actually enter into the space right here in the middle of this pump. And so here we have the potassium inside there. It will dephosphorylate and the phosphate group will leave. And at that point, the pump actually changes shape and turns into this inverted V, and the potassium is released into the intracellular space. So this is a really great example of primary active transport because we're going to be using that ATP to change the shape of a protein, and in the process it will move ions across the membrane. So first here we have sodium is going to be higher in concentration on the intercellular space, and so it enters into the space here in the middle on the first step. When ATP comes in, it will change shape and release that sodium into the extracellular fluid, and at the same time potassium will come in. And then once it is dephosphorylated, potassium is released onto the other side. So as a result, this pump maintains an electrochemical gradient that's really important for excitable cells like muscle and nerve cells to function normally, and it's also really important for body cells to maintain their normal fluid volume. So we'll talk about the excitable cells a little bit later as well. Secondary active transport is driven by the concentration gradient that's created in primary active transport. So we have sodium and hydrogen gradients that are created by the primary active transport that we just saw in that last slide. And this results in kind of a stored energy that can be used to move other substances across the membrane against their own chemical gradients or concentration gradients. So in secondary active transport, you have a carrier protein simultaneously binds to sodium and another substance, and then it changes its shape so that both substances cross the membrane at the same time. We do have a couple of different kinds of these carrier proteins. We have antiporters and symporters. So symporters will actually move substances in the same direction. So here you can see you have the sodium gradient going this way, so you have a higher concentration in the extracellular fluid than the intracellular fluid. And in symporters, you have sodium moves through the channel going one way, and the other substance, in this case glucose, moves in the same direction. Same with here with the amino acid. So those are symporters, sim meaning same. And then the other kind are antiporters, and antiporters move substances in opposite directions. So you're going to still use that sodium gradient that we created with primary active transport, and we use that as the energy to move another substance across the membrane, in this case calcium, moving in the opposite direction because it is an antiporter carrier protein, and here we also have hydrogen being moved across using an antiporter membrane protein. Okay, so we are going to start talking about another type of active transport, which is vesicular or active transport within vesicles. So a vesicle is just defined as a small spherical sac. There's a variety of substances that are transported in vesicles from one structure to another within cells. So they also import materials from the extracellular fluid, and they can also release materials back into the extracellular fluid. So there's a couple different types of vesicular transport that we're going to talk about. We're going to talk about two terms right quick. So first we're going to talk about endocytosis. So endocytosis is when materials are moved into the cells in a vesicle that's formed from the plasma membrane. The opposite of this is going to be exocytosis. And exocytosis is where materials are moved out of the cell by the fusion with the plasma membrane of vesicles that are formed within the cells. And both of these types of transport within cells both need ATP. Remember, because they are forms of active transport. Now, there are three different types of endocytosis that I want to quickly cover. We will go over each of these a little bit. So the first one is going to be the one that we talked about on this slide, and that is receptor-mediated endocytosis. So receptor-mediated endocytosis. The next one is going to be phagocytosis. And then the last one is going to be called pinocytosis, also known as bulk phase endocytosis. And we will talk about each of these in the next few slides. So in receptor-mediated endocytosis, the one on this slide here, the cells have these very specific receptors on their plasma membrane, and they will bind to very specific ligands. And once they are bound, then they're going to actually invaginate the plasma membrane and kind of endocytose into this vesicle here. Then we have this vesicle that's going to kind of bind or fuse with a couple other types of vesicles in the cell, but really the end part here is that we're going to transport it where it needs to go. It's going to actually bind together with a lysosome, and lysosomes will actually have enzymes within them that digest proteins. So here we're going to have that degradation of the proteins within those vesicles. And then once we have that digested material, the smaller proteins, the chemicals and molecules, it's all broken down into its smaller bits, then the cell can actually use that, and these proteins and enzymes and molecules are all going to go other places in the cell wherever they're needed. So that is going to be receptor-mediated endocytosis. So those byproducts are going to be used for creating energy or for use wherever it's needed in the cell. Phagocytosis. This is another form of endocytosis in which the cell engulfs large solid particles, such as whole bacteria or viruses or smaller pieces of cells. There's only a few cell types, specifically phagocytes, that are able to carry out phagocytosis. So this is going to be specifically a phagocyte. It's a specific type of cell. And this is when you have a particle that binds to plasma membrane receptors. So you can see here. And this causes the phagocyte to extend out these pseudopods, or they're called, pseudopods means false feet. False feet. And it kind of like reaches out its little fingers, little feet, to kind of engulf that particle or that microbe there. In most cases, the undigested materials in the phagosome, so once it creates this little vesicle, it's called a phagosome because it's within a phagocyte. A lot of times these remain indefinitely in a vesicle, and it's called this residual body. So whatever can't be digested or can't be used by the cell is left in a vesicle. That's called a residual body. But just like in the last slide, the cell will, or the vesicle will bind and fuse with a lysosome, so you see here, and then the digestion enzymes within that lysosome will kind of degrade that microbe or that particle as much as possible, and whatever is not digested is left within this residual body, but it tries to use as much as possible. So in pinocytosis, also called bulk phase endocytosis, this is where tiny droplets of extracellular fluid are taken up by the cell. This is like when liquid, because it's when liquid is taken up by the cell, it's also kind of a form of drinking by the cell. So no receptor proteins are involved in this process, and all the solutes that are dissolved in the extracellular fluid are brought into the cell. Otherwise, it's pretty well the same process. We're going to have this formation of a vesicle, that endocytosis there. The fluid and dissolved solutes move within the vesicle throughout the cell. They fuse with the lysosome. Everything that's in there is digested by those enzymes, and then the cell uses whatever they can use out of those digested products. So the three types that we have are going to be the receptor-mediated endocytosis, so that's the intake of a variety of substances by receptors on the outside surface of the cell. We have phagocytosis, which are going to be the cell engulfing large solid particles. Usually only phagocytes can perform this action. And then the third type is going to be pinocytosis, or otherwise known as cell drinking. Okay, a couple more terms here for you. Remember that exocytosis, this is when the cells fuse with the plasma membrane and they release their contents into the extracellular fluid. Transcytosis, this is when the vesicles undergo endocytosis on one side of the cell, they move across the entire length of the cell, and then they undergo exocytosis. So this occurs most often across like endothelial cells that line blood vessels, and it's really a means for material to move between blood plasma and the interstitial fluid. So it's kind of like we have here. So this is like a blood vessel, for example. I'm making the lining of the blood vessel very thick, but just to kind of show how it would look. So here we would have endocytosis of the vesicle. The vesicle would move across the cell, and then here we would have exocytosis. So then we have particles. If we're looking at particles within the cell here, we would have these particles here inside the vesicle, and then here they are being expelled from that vesicle. This type of endocytosis and exocytosis, or transcytosis, this also occurs in pregnancy when some of the women's antibodies cross the placenta into the fetal circulation, and this is done by transcytosis. Here is a table, just kind of comparing those different transport types, just kind of to bring everything back together for you, give you a nice little summary there. Additional more summary of the active processes that we talked about. So you can stop and read those if you would like. And that will be the end of part 4.